Good morning friends, my name is Dr. Ankur and I welcome you all on my channel Biomanic. Today I will discuss a very important chapter regarding the board examinations as well as the competitive profiles or the competitive exams. The chapter is photosynthesis. Friends, I have divided this chapter into 5 parts and today I will be discussing the part 1 of this chapter, the photosynthesis part 1. In this part 1 we will be considering or we will be dealing with some very important topics like the very first topic is historical background or the historical importance of this chapter, the different scientists, their approaches, their theories, their explanations, what they discovered. The second is most important and most confusing, especially for NEET students, the pigment system or the pigment molecules. Okay, after that the third subtopic will be photosystem 1 and photosystem 2. Okay, fourth topic will be cyclic and non-cyclic photophosphorylation. Okay, and that comes under Z scheme. Okay, other topic specifically for NEET students, fluorescence and phosphorescence. The same comes under or we discuss bioluminescence or chemiluminescence. Okay friends, we'll start with the same. The very first topic is historical background and all these topics. Okay, a big list. So all these topics except this one, this is the common, it's a starting, is the preface of the chapter. Okay, so all these topics whatever we will discuss, they come under a different specific phase of photosynthesis, and that phase is what? And that phase is the light dependent phase. So the very first point is that we have to justify, we have to prove that during photosynthesis only light and the importance of light is not actually overtaken. It is not actually important, 100% important. It is not only required for the formation of food. Okay, so scientists, they have different scientists. For example, if I will carry on with the topic of historical background, so I will discuss friends, I will discuss about Immersion and Arnold. Immersion and Arnold, they were the scientists. They actually they have conducted a flashlight experiment, and that flashlight experiment, they actually prove, or they came to know that the light having intermittent intensities Intermittent intensities means light phase, then without light, then light, then without light, then light, then without light, then light, then without light, like that. Okay, इन्होंने basically क्या किया, what they have done actually, ठीक है, I will go bilingually for certain explanations. Okay, इन्होंने बेसिकली क्या किया कि इन्होंने एक ऐसा एक्सपेरिमेंट कंडक्ट किया जिस एक्सपेरिमेंट में इन्होंने, ठीक है ना, लोअर प्लांट्स ले लिए जैसे एल्गे ले लिया और उनके ऊपर जो लाइट एक्सपेरिमेंट किया वो फ्लैशिंग लाइट एक्सपेरिमेंट किया. मतलब लाइट पड़ती थी फिर थोड़ी देर के लिए बंद हो जाती थी, फिर लाइट पड़ती थी फिर थोड़ी देर के लिए बंद हो जाती थी. वॉट दे हैव कंक्लूडेड फ्रॉम दैट, ओके, वॉट दे एक्चुअली हैव कंक्लूडेड फ्रॉम दैट the process of photosynthesis includes two phases, the light phase and the dark phase. The light phase is light dependent phase and the dark phase is light independent phase. Okay, as we all have discussed at you studying in class 12, you already know that the site of photosynthesis is basically the plastid or the chloroplast of the leaf. Leaf contains various cells, mesophyll cells, and these mesophyll cells contain what? Chloroplast. And these chloroplast contains what? Thylakoids. Okay, and these thylakoids, if we talk about thylakoids, they are the double membranous structure, and the inner membrane invaginates to form these sac-like structures, and these sac-like structures are known as what? Thylakoids. Okay, the matrix which contains enzymes which are the matrix which contains enzymes, this is outer plastidial membrane, this is inner plastidial membrane, okay, this is thylakoid, this stack or pile of coins is known as what? Grana. इसको हम क्या बोलते हैं? Grana. This intergranal thylakoids, intergranal thylakoids or lamellar thylakoids. Intergranal thylakoids. Okay, now specifically talking about the NEET profile or the NEET students, one or two important anatomical features what you have to remember that, स्पेशली अगर हम बात करें नीट स्टूडेंट्स की तो उनके लिए कुछ थोड़ी सी जानकारी और देना चाहूँगा कि द थाइलाकॉइड मेम्ब्रेन, द थाइलाकॉइड, द इनर पोर्शन ऑफ द थाइलाकॉइड इज नोन एज व्हाट? लॉक्यूलस. इट्स लॉक्यूलस. अपॉलॉजीज फॉर द स्पेलिंग मिस्टेक्स. ओके, the inner space of the thylakoid is known as what? Loculus. Now the thylakoids, they are bunch of coins, they are piles, I mean they are like that, stacks. Okay, so there are two parts where two membrane approaches each other, like this membrane and this membrane, where two membrane approaches each other, that is known as what? That is known as appressed thylakoids or appressed part of thylakoids. Okay, appressed part of thylakoid. Okay, and the other part which is not approaching each other, opposite side, it is known as what? Non-appressed part of thylakoid. Okay, so appressed part of thylakoid, non-appressed part of thylakoid. Why I am telling you? Because they are the site for the distribution of different types of photosystems. Getting my point? ये जो appressed और non-appressed parts हैं, they are the site for the different types of photosystems. Okay, now we are having granal thylakoids, stromal thylakoids. This is granal thylakoid and other are stromal thylakoids. Okay, then intergranal thylakoids. Okay, outer plastidial membrane, interplastidial space, inner plastidial membrane. Okay, so this is the overlook on the anatomy of this thylakoid. Now coming to the historical background back again. So Emerson and Arnold, they have basically done what? They have conducted flashlight experiment. Then coming to Reuben and Kamen, Hassel, Kamen, Then these the group of scientists, they have discovered that, or they have proved that oxygen is liberated from water. During the reaction of photosynthesis, oxygen is liberated from water. For that, they have taken a radioisotope of oxygen. What they have taken? They have taken a radioisotope of oxygen, and that radioisotope of oxygen is taken in the form of water molecule. So that water molecule basically oxidizes itself to form oxygen gas. Okay, the simple reaction is the H2O, this is O18. Okay, this H2O gets oxidized. How it gets oxidized? Because we are having certain H plus acceptors or proton acceptors. We are having certain proton acceptors. For example, like that, we are having a compound A, a molecule A. If this molecule A is a proton acceptor, like for example, we will dealing with the proton acceptors in this reaction, in the coming reactions as well, that this A is NADP, nicotinamide adenine dinucleotide phosphate. Okay, so Vishnik and Onak, they were the scientists that they discovered that during the light reaction, the natural H plus acceptor is what? NADP plus. Okay, Vishnik and Onak. Okay, Rubin and Kamin, what they have discovered? They have discovered that this oxygen is liberated in the form of oxygen gas. It is a non-radioactive isotope, heavy isotope of oxygen. Okay, so we have discussed Emerson and Arnold, we have discussed Rubin and Kamin, we have discussed Vishnik and Onak. Okay, now coming to Van Neal, he basically discovered, what he proposed, that if we take certain proton acceptors, and conduct experiment specifically in lower plants. Okay, so if we take certain proton acceptors, like for example, the experiment of Van Neal was not on algae, actually it was on iron sulfur bacteria. So the iron sulfur bacteria, instead of oxygen, sulfur molecule or the sulfur globule is liberated. Instead of oxygen, the sulfur globule is liberated, because they are sulfur containing bacteria, and in that sulfur containing bacteria, the sulfur is liberated. For example, if I am having H2S, getting my point, friends, H2S instead of H2O, and I am having a proton acceptor over here, so this proton acceptor will gain this H to form free sulfur globule. Okay, so this H2S liberates sulfur to form HA or AH2, whatever it is. So this is what he came to know, that the photosynthesis is oxidative reductive reaction. It is an oxidation as well as reduction reaction. Okay, then Van Neal, this was the Van Neal. Now coming to Robin Hill proposed, he proposed the pathway of photosynthesis, the pathway of light dependent phase. That's why the light dependent phase is also known as Hill's reaction. Okay, he actually conducted experiment on Stellaria media, a kind of algae. Okay, so that experiment was very unique. That experiment was very specific. Okay, so we will discussing about the Robin Hill's experiment, what his experiment is. According to his experiment, he chosen some proton acceptors like chlorophenol, indophenol, like chromates, like ferricinates, like that. Okay, like leaf extract. So in the presence of all these extracts, if we add water along with it, the water will be oxidizing in the presence of sunlight, and this hydrogen will be taken by all these compounds, by all these proton acceptors, by all these hydrogen acceptors. Okay, so Robin Hill, Van Neal, they all came to a single conclusion that during photosynthesis, water is oxidizing itself and CO2 is reducing itself. Water is oxidizing itself and CO2 is reducing itself. Now taking further on the same topic. So this reaction is a kind of redox reaction. Okay. Now coming to the second point that we have to discuss, the different types of what we can say photosystems or different types of pigment systems. Okay. So the reaction is being divided into light dependent phase and light independent phase. The very first step in the light dependent phase is what? Photolysis of water. What exactly photolysis of water means? The thylakoid, the locus of thylakoid or the lumen of thylakoid, we can say the lumen of the thylakoid, it is having what? The pigment molecules. And all these pigment molecules, they are having a specific wavelength to absorb, because all these pigment molecules imparts different colors. Getting my point? They impart different colors. And as we have studied in atomic structure, that this wavelength or this color is being exhibited because of the property of having different types of frequency or different types of energy. Okay. So the energy, what they are absorbing, and the wavelength, what they are emitting, is different. The energy, what they are absorbing, and the wavelength, what they are emitting, is different. As we have studied about photons, the quantum mechanical model, the quantum science, the light, it is having photons. All these photons strike with the pigment molecules. The pigment molecules, they are cluster of pigment molecules, and all these cluster of pigment molecules, they form what? They form a harvesting complex. They harvest the electron, it resonate, then it make the other electron to resonate, then it make the third electron to resonate, like that. Okay. So they form a light harvesting complex. And in this light harvesting complex, all these pigment molecules, they are jumbled, they are assembled to give the electrons. Okay. Coming to the pigment part, what exactly we have to study regarding our exams and all. The pigments, there are basically two types of pigments, which are mostly important. Okay. When we talk about higher plants and green plants, okay, so we are having what? The chlorophyll and the carotene. Chlorophyll and carotenoids, these are the important pigments. The chlorophyll, it is of further various types, like chlorophyll A, B, C, D, E, having different types of formulas. Okay, quite cumbersome to remember the formulas of chlorophyll. So chlorophyll A, chlorophyll B, then carotenoids. See, what we have done a experiment. That experiment you also know that in chemistry what we study, chromatography. So in chromatography, during chromatography, because of the solubility of these different types of pigments based on petroleum ether or dimethyl ether or any organic solvent, okay, these pigment molecules are separated, and we came to know that we are having different types of pigment molecules. So in these pigment molecules, the very first is chlorophyll, okay, like the formula C55, H74, N5, O4, Mg, like that and all. Okay, so chlorophyll A is having different formula, chlorophyll B is having different formula. In chlorophyll A and chlorophyll B, the main difference, is that chlorophyll A is having a methyl group on the third pyrrole ring carbon. Okay, and in chlorophyll B, it is having the aldehyde group on the third pyrrole ring carbon. So we are having different types of structural differences as well. Okay, now coming to the percentage, if we talk about that, chlorophyll A is maximally found. Okay, so in case of different types of photosystems, if we talk about different types of photosystems we are having, how we came to know that we are having different types of photosystems. So these photosystems are basically because that different scientists, they have conducted experiments regarding that the very important experiment is, you also have studied that why we require different types of wavelengths. Okay, you have studied about the action spectra and the absorption spectra, and the other thing we will also be discussing about the immersion red drop experiment. Okay, scientists, they have basically taken monochromatic lights of different types of wavelength, and all these monochromatic lights of different types of wavelength, Monochromatic light, they focused on different types of having different types of wavelength. So they came to know that because of having different wavelengths, the energy absorbed by the plants and the light used, what the light they absorbed and what the light they used, okay, they are having different types of ratios. Mean the absorption part is different and the action part is different, and this makes the basis of action spectra and absorption spectra. Okay, for example, we are having different types of pigments in the light harvesting complex, like chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E, okay, beta carotenoids, carotenes. Okay, we will be discussing for the same. Okay, so all these pigment molecules, they form a light harvesting complex, and all these pigments molecules, they take different types of wavelength, that is, they are having different type of energy, and because of having this different type of energy, they perform different function in the same wavelength, in the particular wavelength. That's why their action spectrum is different what they are showing in their absorption spectra. ठीक है ना, ये जितने भी wavelengths different pigments के हैं, क्यों different है? क्योंकि उनके एनर्जी एब्जॉर्व करने के जो रेशियोज हैं वो डिफरेंट है, तो वो अलग अलग एनर्जी एब्जॉर्व करते हैं, इसी तरीके से वो अलग अलग काम करते हैं उस पर्टिकुलर वेवलेंथ में, तो जो उनका एक्शन स्पेक्ट्रम कहलाता है। और एब्जॉर्प्शन स्पेक्ट्रम का मतलब उन्होंने क्या लाइट एब्जॉर्व की, ठीक है ना, किस किस वेवलेंथ की लाइट को एब्जॉर्व किया और उस वेवलेंथ की पर्टिकुलर लाइट में उन्होंने क्या काम किया, तो ये दोनों चीज़ें हमें देखनी पड़ेंगी। ठीक है ना, तो चलिए फ्रेंड्स, ये आज हमारे पास प्रीफेस था इस टॉपिक का। ठीक है, I have discussed the preface of this topic, the historical background and the different types of pigment systems, and in these pigment systems I will be discussing more regarding for NEET students. Okay, so for this time I'll take your leave. Thank you.